Mitosis versus Meiosis by Peter Phillips and Zane. Mitosis is complex and highly regulated. It is when a eukaryotic cell separates the chromosomes in its cell nucleus into two identical sets in two daughter nuclei. Interphase is a phase in the cell cycle in which the cell spends a majority of its time and prepares for cell division. It increases its size and number of organelles and makes a copy of its DNA. In G1 phase, the cell increases in mass. In the S phase, the DNA is synthesized. And in the G2 phase, the cell synthesizes proteins and continues to increase in size. In the latter part of interphase, nucleoli are present. The cell's chromosomes have duplicated and are in the form of chromatin. There are four phases in mitosis. The first is prophase. In prophase, the chromatin condenses into discrete chromosomes. The nuclear envelope breaks down and spindles form at opposite poles of the cell. The next phase is metaphase. In metaphase, the chromosomes are aligned at the metaphase plate, which is equally distant from both spindle poles. The third phase is anaphase. In anaphase, the paired chromosomes or sister chromatids move to opposite ends of the cell. The last phase is telophase, in which the chromosomes are condoned off in distinct new nuclei in the emerging daughter cells. Cytokinesis is also occurring at this time. Cytokinesis is when the cytoplasm divides. It is in the late stages of metaphase and sometimes meiosis, splitting binucleate cells in two to ensure chromosome number is maintained. It also differs in plants and animals. At the end of mitosis, two distinct cells with identical genetic material is produced. The importance of mitosis is the maintenance of the chromosomal set. Each cell formed receives chromosomes that are alike in composition and are equal in number to the chromosomes of the parent cell.
Meiosis is a two-part cell division process in organisms that sexually reproduce. Before a cell enters meiosis, it must go through interphase, which is where the cell grows and synthesizes DNA. In prophase 1, chromosomes condense and attach to the nuclear envelope. Also, synapsis occurs and a tetrad is formed in which each tetrad is composed of 4 chromatids. Prophase 1 is where crossing over may occur, unlike in prophase 2. Chromosomes thicken and detach from the nuclear envelope. In metaphase 1, tetrads align at the metaphase plate and centromeres of homologous chromosomes are oriented towards the opposite cell poles. In anaphase 1, chromosomes move to the opposite cell poles. The microtubules and kinetochore fibers interact to cause the movement. The homologous chromosomes move to opposite poles, yet the sister chromatids remain together. In telophase 1, the spindles continue to move the homologous chromosomes to the poles. Once this is complete, each pole has a haploid number of chromosomes. In most cases, cytokinesis occurs at the same time as telophase 1. At the end, 2 daughter cells are produced, each with 1/2 the number of chromosomes of the original parent cell. The differences between meiosis 1 and meiosis 2 is that in meiosis 1 there is presence of crossing over. In meiosis 1, homologous chromosomes move to opposite poles as sister chromatids remain attached, but in meiosis 2, sister chromatids separate. In meiosis 1, chromosome complement is reduced from a diploid to a haploid, yet meiosis 2 is a mitotic division of haploid cells produced from meiosis 1. And finally, there is no interphase between meiosis 1 and meiosis 2. In prophase 2, the nuclear membrane and nuclei break up while the spindle network appears. Chromosomes do not replicate any further in this phase of meiosis. In metaphase 2, the chromosomes line up at the metaphase 2 plate at the cell center. The kinetochores of the sister chromatids point toward opposite poles. In anaphase 2, the sister chromatids separate and move toward the opposite cell poles. In telophase 2, distinct nuclei form at the opposite poles and cytokinesis occurs. At the end, there are 4 daughter cells, each with 1/2 the number of chromosomes of the original parent cell.
Mitosis produces 2 diploid cells, while meiosis produces 4 haploid cells. The purpose of mitosis is to produce new body cells and does not allow for genetic variation, while meiosis allows genetic variation through multiple processes. Meiosis produces sex cells, which are key in reproduction of multicellular organisms. Each new cell contains half the number of chromosomes. Meiosis has 2 cellular divisions, while mitosis has 1. However, they are both necessary to support life, and they both produce diploid cells in the first division.